Hello, good morning to all.
Good morning, sir.
Hi, sir. Good morning, Agatha.
Good morning, Edric.
Good morning to all. Very good morning, guys. How are you all?
Good, sir.
Good, sir.
Okay. So everyone turn on the camera so that we can start our classes. And we will directly start heredity chapter. Heredity is 23 now?
Homework?
Yes?
You checked our homework, right?
No, I didn't check it. I was busy whole day from yesterday. Today I will check both the homework. Yesterday's homework as well as day before yesterday homework. And on Monday we can take your doubt on that. Okay?
Sir, today is a carbon test.
Yeah, I will send that carbon test after the class. Only to you. And then everyone I will send on Saturday. Okay.
Why for them Saturday? But Saturday is a weekend, sir.
Yeah, so you can give on with Friday, no?
Today, right?
Yeah. So what chapter we were doing? Chapter number?
25. Gata. Gata. Gata.
Okay.
Gata.
Heredity we were doing and we did that capital T is the dominant allele and small t is recessive allele. Yes?
Yes, sir.
So
Yes, sir.
now the first law was law of dominance. Law of dominance which says that dominant allele dominant allele shows the expression in heterozygous condition. Which condition? Heterozygous condition. Now what is heterozygous condition? Yes, anybody?
Yes, sir. So heterozygous condition is a condition in which it has both dominant and recessive alleles.
Yeah, it has both dominant and recessive allele. So for example capital R small r. So in this the capital R is dominant and small r is recessive. Understood, David? Miraculin, turn on your camera, please. You got it, Agatha?
Yes, sir.
Okay. This is called as dominant allele. This is law of dominance. Now, second is law of segregation. Law of segregation. Law of segregation says that law of segregation says that during during gamete formation, allele divides allele divides segregately, or we can say allele divides independently. Independently. This is the second law. See, we'll understand this. Everyone, please pay attention. I will let you understand this. Okay, for example, we have this chromosome, this circle, which is a cell, and the cell has chromosome. So I will make chromosome, and each chromosome has one allele in it. So I told you, like for example, capital T and small t. Understood? Agatha, David, you got it? Okay, it's gonna be look like this. Okay, now when division will come, when division part will come, then you know what will happen, that this strand and this strand, this strand which is capital T, this strand which is small t. They segregate independently like this. This is an example of law of segregation. Now, from this, when two gametes will form, so it gonna be look like this. Like this. Everyone got this?
Yes, sir.
Agatha, David, Ira, Miraculin, you got it? This is called as law of segregation. That when the allele or when the chromosomes divides in gamete formation, they divide independently. They do not divide on each other, but they divide independently. Hence, it is called as law of segregation. What it is called? Law of segregation. So, so for example, the factor of tallness in the pea plant always expressed in the offspring, but dwarfness expressed only its factor of tallness is not present. So, what does this statement is trying to say? When small t and small t is there, then we can see the dwarfness of the plant. So, Agatha, why only small t we can see the dwarfness? Why not in capital T and small t?
Because small t, because there's no dominant, because small t, because both are small t, and small t says to make the plant small.
Yeah, why not in capital T, small t?
Capital T is more stronger than small t, and capital T wants the plant to be big, and the small t wants the plant to be small. So the plant will be tall.
Yeah, because dominant character. The simple answer is dominant character. Okay, understood? Okay. Now there is also third law, which is called as law of independent assortment. It is called as law of independent assortment. So now they didn't write it, but for our information, we can say that like right now we are taking how many character? Agatha, how many character we took? Only one character,
Only
which is
two, right?
no,
Capital T
no,
and small.
that is two parts.
Ah, yeah, yeah.
But character is only one, height.
Yes.
Height. Okay, height character. Okay, Edric, how many character we took? Only one character we took. It is called as monohybrid cross. Okay, because we take only one character. So these two laws can be explained in monohybrid cross. Which cross? Monohybrid cross. Okay, but we have one more point which is called as dihybrid cross. Dihybrid cross. Dihybrid cross means di means two, hybrid means character. So when we take two character and then we cross it, it is called as dihybrid cross. Are you getting my point? No, you're not getting my point. David, ask me.
Yeah, I got it.
Okay. So dihybrid cross means when we take two character. How many characters? When we take two characters, it is called as dihybrid cross. Now, how dihybrid cross look like? For example, I took two character of a pea plant. I take two character of a pea plant. First is round yellow. Round look like this, and it is in the yellow color. Okay, and the second one is wrinkled green. So wrinkled green look like this. This is wrinkled like pea plant. This is wrinkled and it is green in color. It is called as wrinkled wrinkled green. This is round yellow. This is wrinkled green. Are you getting my point? Yes or no? So round is the pea plant seed, like the pea. Okay, that is round. And yellow is yellow in color. In the same way, wrinkled is the outer covering of a pea plant, which is wrinkled right now. And green means green in color. Are you getting my point? Yes or no? Round yellow, you can write it by capital R, capital R, capital Y, capital Y. This is called as round, and this is called as yellow.
So is this based on the law of independent assortment?
Yeah, I'm trying to explain law of independent assortment little, not going into too deep. Just little part I will touch.
Okay.
Okay, and this is called as wrinkled, which is small, written as small r, and green, which is written as small y. So you can now assume which one is the dominant, which one is recessive. Agatha, which one is dominant?
Round yellow.
Round yellow is dominant. Okay, and David, which one is recessive? Say loudly.
Wrinkled green.
Okay, wrinkled green. Yes. So now we took two characters, and when we cross these two characters like this, so these two characters were independently, like it is called as law of independent assortment. Now they will independently work on the chromosome. So it's gonna be look like this, like they have a chromosome. Now this strand has capital R, and the another which is capital Y. This strand also have capital R. This strand is also a capital Y. It's gonna look like this. Okay, if I will make a chromosome of this, so this will have small r, this will have small y, this will have small r, and this will have small y. So what I'm trying to say, when we in a dihybrid cross, when two characters is taken, they work independently. Means they will not depend upon the other character. Like yellow color is not dependent on the round character. Are you getting my point? No. Round and yellow, they will independently work. They will independently assort when the division will come, when they will combine. So they are not dependent on each other. This is called as law of independent assortment. Are you getting my point, everyone?
Like, what do you mean they're not independent of, like, what do you mean by independent?
Means round character will not disturb the yellow color of the seed. It's gonna be look like this. Like round character came and then the yellow become purple, it won't be like this. Got my point? No.
Yes,
So,
sir.
like, the round character will not change the yellow character.
Yeah, both the character will work independently. They are not disturbing each other. They will not disturb each other.
But a capital R and a capital, but a round yellow will overtake the wrinkled green, right?
Yeah, will work as a recessive, like will work over the recessive, like round and wrinkled. Which one is dominant? Round. So when the situation of capital R, small r will come, so round seed will be there because dominant character is there. But round and yellow is there. So for example, yellow seed will always round. It won't be like this. Okay. Like if the situation arises like small r, capital Y, so it will become wrinkled green. It will become wrinkled green, not round green. Got my point? Yes or no?
Yes, sir.
Agatha?
Yes, sir.
Yeah, keep your camera in the center, no? So now I will give the this thing uh, law. So you guys just write it down, okay?
Yo.
Okay, we have this thing.
Do I need to write all of them?
Yes.
Okay. Give me 10 minutes.
So can you write the law of dihybrid cross?
You want no, there is no law of dihybrid clause. There is
Yeah.
meaning of di.
Yeah, definition.
meaning- Yeah, that okay, I will write it down. Huh? Huh? Everyone got this? No. Agatha at Hawaiian Beach Bridge. You got this?
It's not Hawaiian Bridge, it's Golden Gate Bridge.
I mean, yeah, whatever the bridge is there. My geology is very bad.
It's not geology, it's geography.
Yeah, that is why it's very bad. Okay. You guys write it down. I'm coming in 2 minutes. Okay.
Edric,
Yes.
can you explain me the term like supposification in the
Huh?
like that SAPOFICATION?
Saponification.
Yeah.
Okay. So saponification, no. It's basically the reverse of esterification. So esterification is a process in which the ethanol and ethanoic acid are mixed together to form ester, which is called ethyl ethanoate. So ethanol and ethanoic acid will form an ester. But saponification is the reverse. That ester will be reversed back into ethanol and ethanoic acid. So the conversion of an ester into ethanol and ethanoic acid is called as saponification.
Thank you.
Welcome.
Edric, I had a question. How do you zoom in without using your scroll, like mouse scroll wheel?
I have a math mouse. The mouse is called MX Master 3S, and I like the middle, like I have the scrolling. Like, I just zoom in and zoom.
Mine's not working. I have this mouse, but the scroll wheel is not working.
For me, it does like.
Barely works.
For me, it works properly because the mouse is called MX Master 3S.
Can you change the clicks for?
Okay. Done?
Not yet, sir. 2 minutes.
We write it down. David, what happened to your mic? No, I'm just asking what happened. It's always have disturbance.
So I don't know. I was trying to figure that out.
It's always have that grrr sound, like disturbance, some disturbance there. Yes, Agatha, you're saying something.
No, I was just saying that I'm done.
Oh, so you got this three laws: law of dominant, law of segregation, and law of independent assortment.
Done, sir. Got it.
Okay. Now we'll go to the next part, which is chromosomes and genes. So we are not talking about any genes like Wrangler or Levi's, any genes. We are just talking about the genes, the chromosome genes. Sorry for that. Okay. So chromosome. The nucleus of every cell except RBCs of mammals contains a fixed number of chromosomes. In all cells, or eukaryotes, chromosomes shows the following typical characters. Eukaryotes are basically called as RBCs. Okay. No. Eukaryotes are not RBCs. Eukaryotes are the those cells which are highly advanced. That is called as eukaryotes. Okay. They are present in they are they are present in pair, one from father and one from mother. So as I told you, I told you that during segregation, two strands are there. So when gamete formation is there, two strands are there. Okay, I will tell you this thing again. Okay, everyone, please pay attention. So we will understand it like this. Okay, so I told you that male has 40. Basically, it is in the like this. 22 pair of chromosome plus XY. This is male. This is male. Okay. Male has 22 pair of chromosome. Pair means into 2. Okay. Pair means into 2. So basically, how much it will become? 44 plus XY. Got my point?
So
Yes
44
or no?
plus 2 is 46, right?
Yeah, basically 46, in which 22 pair, which is 44, is basically called as autosome, which decide the height, character, and all these things. Okay. And the XY is called as sex chromosome. Sex chromosome. Autosomes and sex chromosome. Sex chromosome determine the gender, the sex of a child. If XY is there, it is called as male. If XX is there, it is called as female. Okay. You got my point, everyone?
Yes, sir. So,
Okay. So in the female also, there is 22 pairs of chromosome plus X and X. 22, which is called as 44, is basically called as autosomes, and XX is called as sex chromosome, which determine the sex. Okay, now everyone please pay attention. So what will happen when gamete formation is there? So how many chromosomes they have? Basically 44 plus XY. 44 plus XX gonna be look like this. This is this is. Now it will create sperm, and this will create ova. Okay. Now in the nucleus, it has how many chromosome? 44 plus XY. So what will happen? That these two strands will divide and come, one strand will come this side and one strand will come this side, like gonna be look like this. Are you getting my point?
Yes, sir.
Such that this will have 22 plus x and this will have 22 plus y. Everyone understood?
Yes,
Yes
sir.
or no? No.
Yes, sir.
Okay. In the same way, the female has like this. Okay, it has 44 plus xx. So in this, one strand will have 22 plus x and this strand will also have 22 plus x. Everyone got it? Understood? Now, this is called as gamete formation, with the number of chromosomes become half. Okay, this is called this process in which the number of chromosomes become half. It is called as meiosis. Meiosis. So, what is the meiosis? Meiosis is the division of a cell in which the number of chromosomes becomes half. Everyone got it? Yes or no?
Yes, sir.
Okay, one more thing.
Why,
Now,
why?
this situation in which the number of chromosome is double, like 2n condition, it is called as diploid stage. It is called as what?
When is the
Diploid stage.
chromosomes doubled?
Like when it is like here it is half, no, and here it is double than the half. For example, here it is 44 plus xy. Here it is 22 plus x. So when it is called as diploid stage. So right now, if I will take out the hand cell, or hand like cell in my hand, and if I will examine it, it is in the form of diploid cell. Means 44 plus xy. Okay, and when it is in the form of gamete formation, it is basically in the N form. It is called as haploid stage. It is called as haploid. So haploid in the gamete formation, when the number of chromosomes become half, it is called as haploid. And diploid is basically the normal situation. You got my point? Yes, Agatha.
So gamete formation is also meiosis.
Yeah, gamete formation goes through meiosis so that the number of chromosomes become half. You got my point? No.
Meiosis is a cell division, right?
Meiosis is a cell division in which the number of chromosomes becomes half.
But
Now, yeah.
But sir, mitosis is also cell division, but there the chromosome will
Remain same.
yeah, chromosome
We will
will.
learn. We will learn. Okay, now everyone please pay attention. Please pay attention. Now, for example, this sperm came to this ova and they fuses. So now it will become two cell stage. That two cell stage is called as zygote. Because two cells are meeting and it become two cell stage. Now inside the zygote, one strand will be there which is from the male. It is called as 22 plus x, and another will also be there. It will also have 22 plus x. Now this zygote will become what? Male or female?
The female.
Yes, it will be female. Why? Because 22 plus 22 will become 44. The chromosome restored and plus XX. And 44 plus XX is basically called as female. This will become female. You got my point, Agatha? No.
Yes, sir.
Okay. Now, for example, this chromosome and this chromosome mate. So it is also become zygote. And in zygote, there are two strands. The one will be have 22 plus y and another one will have 22 plus x. So now this will become what, David? Male or female?
Male.
Yeah, it will become male because it will restore 44 plus xy. So in the end, it will have 44 plus xy and this will have 44 plus XX. This is how the sex determined happened. So why the question can come, why the number of chromosome becomes half during the gamete formation, or why the question can come, why the meiotic division happened during the gamete formation. So it happens so that in zygote the number of chromosome will be should have same as the parent cell. Like parent cell has 22 plus XY, 22 plus XX. So in the same way, the baby, the zygote should also have the same number of chromosome. If it is not goes to the meiotic division, then the number of chromosome will be double. It will be double. Everyone got my point?
Yes, sir.
So write it down now. So this will have 44. No. Write it down.
Ma'am, sir, there's no space for me to write.
Why?
Uh, I, I've written a lot in this class and my other science class.
Take one paper from other book, then write it down. Okay, everyone, done?
So, on March, we will you will start with the new batch. So, between that, is there any holiday or something?
No, it won't be. Like I will complete my course with you guys like 6th of March, 6th or 5th, 7th of March, and 9th of March we'll start with the new batch.
Like only weekend holidays for us,
Yes,
and
yes.
Christmas holidays. That's it.
Yeah, that's it.
So what
Like,
about
yeah, in the month of June, I take holiday in the month of June.
Like,
So
The
let's see.
summer holidays.
Yeah,
so why are summer holidays in June?
yeah, in June. Till
For
then, there
us,
is no.
for us, summer holidays is in April and May.
Yeah,
yeah,
like in the south, April and May, like it is hot, but here it is June. So
for you,
holiday.
Where do you live?
okay. Miraculin lives in Ludhiana. I live in Bangalore.
But my native is Chennai, a place
Oh,
called Ambattur.
okay.
Okay.
So please finish the chapter by today. It's Friday.
Yeah, there, I'm trying my best, beta. I'm trying. Okay, now we'll go through it. So they can seen during the cell division in a non-dividing cell, they appear in the nucleus jumbled in the form of chromatin. So I told you, like DNA is normal a thread form. When some condensed form, it become a chromatin. So I will share you one picture with you guys.
So, but Newsroom doesn't allow to go to YouTube and or Google and search and find a picture. Newsroom doesn't allow that. Only old Zoom allows that.
Which part, beta?
Sir, Newsroom doesn't allow to a search on YouTube or Google for a photo or a video. It only allows you to paste the picture on the whiteboard, the Newsroom.
Yeah.
That's the problem with Newsroom.
Okay. Okay. Now see this picture. Everyone, see this picture. We have this picture in which it is saying that this is basically a DNA form. This is a DNA form. Okay. Now DNA is like they have four type of base: adenine, guanine, thymine, and cytosine. And uracil is also there, but uracil is found in RNA. Okay. So adenine, guanine, thymine, and cytosine. We have four type of bases, and you can see that the green color represent the guanine. The cytosine is purple. So you can see the combination between the strands. This is called a strand. This thread like it is called a strand, and the strand has like two strands are joined by the bond between the bases, nitrogenesis bases of these things. Are you getting my point?
Yes, sir.
Okay. Now, this is basically the strand is made up of sugar phosphate backbone. So the main thing is these bases, the adenine, guanine, thymine, and cytosine. So now these DNA will condense themselves to form a chromosome. And like this one strand, it is called as chromatid. Okay. This
Yeah.
one strand is called as chromatid. So when that division will happen, you know what will happen? From this point, from this point, the centriole is a cell organelle which attach with this one strand, and it will it will like pull that one strand gonna be look like this. So one centriole will pull like this, and the another one, like it gonna be look like this. This one strand will be taken up by the centriole, and the another strand will be taken by the another centriole, and then division will happen and become one cell like this. You got my point?
Sir,
This
is it
called as number of
centriole?
chromosome become half.
Sir,
Yeah.
sir, is it centriole or centromere?
Centriole. Centriole or centromere. Let me check. Centriole. See, centriole act as an organizer, while centromere is the attached point of for chromosome like this is called as centromere. So centriole attach the DNA strand from centromere. Okay.
Oh yeah.
Okay, Edric?
So centriole is the center point of the chromosome, right?
Yeah. Okay. So now we have this. Did you understand how the condensed form happened? Okay. So now we will understand this
I'm
more
not
clearly.
feeling fully.
Yeah. See, the condensed form we have to understand, not the division part. We have to understand the condensed form. How it condenses. You can see this is DNA, and this DNA is now condensing each other to become a chromosome. So that when condensed form of DNA is there, then the easy separation can happen. Easy separation can happen. That's why the condensation happened.
So now the DNA is condensed into chromosomes, right?
Yeah, it condenses into the chromosome, and now these chromosome will divide and becoming one strand each. Gonna be look like this.
So chromosomes are condensed DNA.
Yeah. Okay. Now what will happen? This centriole will attach to the centromere, and this centriole will attach to this centromere. Okay. The next step, it gonna be look like this. And this cell will divide like this. So gonna be look like this.
Is this mitosis or meiosis?
Meiosis, beta, number of chromosomes becoming half. You got it, David?
So
After this, yeah.
meiosis, the other one is what?
Which one?
Meiosis and other one is what?
Mitosis.
What is that?
See, in mitosis, the number of chromosomes remains same. If 44 is there in parent, the child will has also 44. That is called as mitosis. But in meiosis, the number of chromosomes become half. So this will become one sperm, and this will become another sperm. Agatha, understood now?
Yeah.
Okay. Okay. Now we'll talk about the human chromosome. What is human chromosome? Okay, human chromosome is basically there are in diploid number it is 46 chromosome. How 44? 46? 44 plus X and Y, so it become 46. David, did you understand?
Yes, sir.
And in haploid condition, it will become 22 plus X or 22 plus Y. So in haploid condition, basically it's 23 number of chromosome. 23 can, how the 23 can be separated? 22 plus X or 22 plus Y. Got it?
Yes, sir.
Yes. Now remember this thing. The Y chromosome in human male is the smallest chromosome. You can write it down in your copy. Y chromosome in the human male is the smallest chromosome. It is the smallest chromosome. You can write down in your copy. It is the smallest chromosome.
Smallest chromosome in human body.
Yeah.
So that out of 23 pairs of chromosomes, 22 pairs are termed autosomes.
Yeah. Good. Out of 23, 22 is what? Chromosome. Autosome, sorry.
Autosome. The
Just give me a
23rd
second.
pair is called sex.
Yeah, the 23rd, which is X or Y, it is called as sex chromosome. So yeah, we have this thing. You can check that we have this sugar bases and we have this thing bases like adenine, guanine, thymine. These all are the bases, and in which the DNA strand are there, and then it condense and become a chromatid, which is the one like chromosome. Is this is chromosome, and if I will take out only one strand, it is called as chromatid. Okay, this is called as chromatic. Everyone understood? So I request Agatha to read this part, please.
Genes are present on chromosomes. The genes are the Mendelian factors present in pairs, one received from the father, other from the mother on the chromosomes. Thus, one member of a pair of genes present on the chromosomes has its pair on the homologous chromosome at the same location. Genes are the bearers of hereditary characters or the units of the hereditary. It has already been mentioned that a chromosome contains one molecule of the chemical called DNA. Genes present on chromosomes are segments of DNA. Figure 25.5.
So genes are basically the bearers of hereditary characters. Okay, what is bearers of hereditary character means? Means they have the characters that can be transferred. Heredity means they can be transferred to the parent cell. So genes are the functional unit. I told you in the previous classes also. Like a person who is an architect of a house, he has the map, the print, the thing, the what is it? What is it? The map of a house.
Design of the
Okay.
house.
Design,
Design.
the map designing, everything he has. But the workers are basically called as genes. They do the works. Okay. So even though the map is in with the architecture, but the main workers are the genes that helps them to inherit the characters to the next generation. Got it?
So the DNA is like informing the genes on how things should work.
Yeah.
You know?
So
Yes.
genes are the bearer, like they can be transferred.
Yeah, genes, those characters which are transferred from parents to the offspring is through genes, because genes contains the heredity characters that can be transferred. Yes, David.
So is it called blueprints of a house?
Yeah, blueprint. Okay. So now we will understand DNA fingerprinting. So Edric, please read this.
25.3.4 DNA fingerprinting. You might have heard that criminals can now be identified by DNA tests called DNA fingerprinting. This is because DNA of an individual is the same in each and every cell of the body and also resembles the DNA of parents. Needless to say that this is because children inherit DNA from their parents. Just like the fingerprint, DNA of every individual is unique, and even if a hair or drop of blood or semen of the criminal is left at the site of crime, it can be used to detect the DNA of the criminal and compared with that of the suspect to ascertain the truth. Figure 25.6.
Give me a second, no?
This
Again
is
becomes
something recently discovered, right?
sorry.
So
This is something recently discovered, right?
Yeah.
Even a fingerprint can be used, right?
Yes.
Hargobind, Dr. Hargobind was the creator of the man-made gene.
My Zoom get glitch.
It's not Hargobind, it's Hargobind.
Oh, okay. Hargobind.
Okay. So yeah. So DNA fingerprinting is done by the Dr. Hargobind Khurana. Okay. This is basically the DNA fingerprinting. Attract.
Okay, sir.
Okay. Now DNA molecule. DNA molecule is basically a polynucleotide. Okay. Why? Because poly means many and nucleotide means they contain nitrogenases bases, deoxyribonucleic sugar and phosphate bond. Okay. The DNA has these things. Now, what are the nitrogenases bases? Are
So,
called as adenine, guanine,
adenine,
thymine,
guanine,
and
thymine.
cytosine.
Right.
Okay. Cytosine. Okay. And DNA is in the form of double helix character. What is double helix character? So double helix means in the form of like this. Okay. Now DNA replication. This is important thing. So DNA replication, cell division takes place in a manner so that one cell divides into two identical cells with the same number of identical chromosomes. Therefore, prior to the cell division, every chromosome should contain two chromatids made up of identical DNA molecules. This is achieved through the process of DNA duplication or DNA replication. Okay. So how the DNA replication happen? So we will understand it like this. See, I will take why this happens again and again. Okay. So what usually happen in this? You can see it. See, there are basically you can see that this is the structure of DNA, which is in the form of double helix structure. Now each strand, when they are dividing the parental strands, the green one which they are dividing, they will make a replication like this. So in daughter cell, each strand when it is dividing, so this cell will get the replication of it, and this strand will also get the replication of it. So now the daughter cells will have two DNA, like one DNA is there and the replication, the duplication occurs simultaneously. You got my point?
So, like the parent's DNA will split, and the daughter DNA will take half of, and there will be two daughter DNAs who take half of the parent DNAs.
Yeah, and the half of the parent DNA will replicate or duplicate to become the 2 DNA again, because DNA in the form of helix structure, no, only one strand is not enough to survive. He needs 2 strands, so another strand will duplicate or replicate. Okay, so this is called as DNA replication. So DNA polymerase is an enzyme catalyzes the formation of new daughter strand, which can form double helix with one strand of parent DNA molecule. So the one parent DNA molecule that came from parent, that DNA polymerase will help in duplicate to make double strand again. You got my point, Agatha, Miracle, Ira? Everyone got my point?
Can you
Two
explain
identical,
the difference,
yes,
like helix? What's helix and all?
helix structure. This is called as helix structure. This is gonna look like helix structure, like 2 snakes are rolling on each other like that.
Ayyo.
Okay, two identical DNA molecules then come two chromatid, which is attached by a centromere. So centromere, what is centromere? Centromere, like when chromosome is there, this point is called as centromere. When 2 chromatids, like this is one chromatid, this is another chromatid. When 2 chromatids meet together, it is called centromere. Are you getting my point, everyone? Yes or no? David, you understood? Okay. Okay. Blood group and inheritance in human being. Okay, now this is the important thing that we need to understand. Okay, so basically, our body has 4 types of blood group. Basically, not 4, it's more than 4, but we will understand only 4. First is A blood group, second one is B, then AB, and then O. So we have these types of blood group. Okay, now, okay. Now, everyone, please pay attention. This is the another thing that I'm explaining it to you.
Sir,
Yeah,
if the mother has O blood and the father has B blood, then what blood will the baby have?
we will understand. We will get it. No problem. Just give me a second. I'm making this chart for you guys. Okay, we have this thing. Okay. Now, everyone, please pay attention. So, how the blood group get determined? So, first of all, before going into this, we should understand 2 things. First is antigen, and the second one is antibody. Now, everyone, please pay attention. What are antigens and what are antibodies? Now, antigen are basically the bacteria. Okay, like when we got cold, so we get bacteria from outside. Because of that, we get cold. So that is called as antigen. So any bacteria or any foreign particles that comes into our body, that becomes an antigen. And antibody is the basically it is produces by our body to resist the antigen. Produces by our body to resist the antigen. So what will happen? Like antigens are like a thief and antibodies are like police. Everyone got it?
Yes, sir.
Okay. So antigens are the any foreign particles that come inside our body. Okay, so I will ask this question. So, for example, you have means a person having a cold. So he will go and ask for the, like, sometimes if we are not repaired with our own antibody, we take antibody from outside. Like we have different antibodies like azithromycin, like these different antibodies are there. Sometimes if we not get like if we not get good by the time we take antibody. Yes, this is correct thing. So we sometimes we take antibodies from outside also. Now, for example, if a person is having a, a problem in the kidney, will he go for the cold antibody?
No.
No. Why? Because antibodies, like it gonna look like this. The perfect antibodies, like for example, the antigen is in this shape. So the antibody should be perfectly matched with the antigen so that it can catch it. If it is like, for example, this is an antigen, everyone please pay attention. For example, if this is an antigen, so the perfect antibody should be there so that it can kill the antigen. If perfect is not there, okay, so it will not interlock with the antigen and it will not kill the antigen. Are you getting my point?
So different antigens need different antibodies.
Yes. Yes. Like for example, if a cold antigen is there in your body, you need a cold antibody for that to resist the antigen. If it is not there, so if you will take any other antibody that rather than cold, it won't work because cold antigen can be killed by the cold antibody. If your body is not making enough to fight with the cold antigen, if you're taking antibody from outside, you need a perfect antibody for that. Yes, you got it, everyone?
Yes.
Okay.
Fever is
So
also
now,
antigen and antibody, right?
yeah,
In the case of any sickness that also includes fever, there's something like an antigen and an antibody.
antibody. So if you are taking antibody from outside, so it means that you are immunizing yourself from outside. Okay. Now we have four types of blood group. Why I taught this to you, I will explain it now. Now, for example, a person having A blood group and a person have B blood groups. So we have this blood group. This is also blood group. And we have AB blood group. And we have O blood group also. So we have these types of blood group. Okay. So I will write it down this. This stands for blood group, BG. It's blood group. Or shall I write full? I will write full. So I can expand this now. Okay. Now listen. So we have first I will write here blood group. Okay. Blood group. Then I will write antigen. Then I will write antibody. Then I will write donate no receiver. And then I will write donate donation. or donator. Now, now, now we have these types of blood group. First is A,
ABO.
A, then we have B, then we have AB, and then we have O. Now, a person having a blood group. So basically, this A, B, AB, and O. These are basically the antigen present on the RBCs. So on RBCs, if a person having a antigen on RBCs, if a person having a antigen, it has a blood group. Everyone got my point?
Yes, sir. So A antigen means A blood group, B antigen means B blood
Yeah.
group.
If a person on
So
a
is an
RBC,
antigen from, so is an antigen germ from outside the body, not produced from the body?
sorry, say it again.
I thought antigens were bacterial or any foreign particles that come in our body.
Yeah, that is correct. But our body, like a person having a blood group, it automatically has a antigen, which is not harmful for the body. That's why the antibody is not there.
So like when the antigen is coming in, it will change itself to some A antigen, so it can be accepted.
Say it again. Say it again.
So it's like an undercover thief entering
Yeah,
the blood group.
undercover thief, which not harmful, which not giving harm to our body. Simple as that. Okay. So now, if if a person having a B blood group, it has B antigen. If a person having A and B, both antigen on the on the RBCs, it has AB blood group. And if a person doesn't have any antibody, if a person doesn't have any antibody, it is called as any antigen, it is called as O blood group. Everyone got this? Yes or no?
Yes,
So if
sir.
a person
So
having a
O blood
blood
group
group,
has no antigen.
yeah, O blood group doesn't have any antigen. Now everyone please pay attention. Please pay attention. We will understand it. Okay. Now A person is basically having a antibody, and B antigen having B. AB has both A and B, and O has
Zero.
no. So no antigen. I will write here is no.
Or, or,
Now,
or, or put zero. Put a zero mark. Put zero.
yeah, put a zero and then become same like. Now please pay attention. Now if a person having a antigen, so he will have a antibody B. Okay.
Yes.
That is why you can't give a person having a blood group, a person having a blood group, no, B blood group to the person to the A blood group. Because if I will give B blood, like for example, I have B blood group and David has A blood group. If I will give my blood, which is B blood group, to the David, so David have antibody B and my blood group is B and it has antigen B. Both will fight and they will die. You got my point? No. Shall I explain it again?
Yeah. No, sir. No, sir. No, sir.
So
Got
like
it.
the antibody of your blood group will so like the antibodies will die, right? And
Yeah.
antigen.
Yeah, the antibody will die. So
Is there
basically,
any
B
use for
blood
the
group.
antigen?
Sorry.
Antigens are harmful, right?
Yeah, antigens are harmful, but these antigens are not harmful. These
No.
antigens are not harmful. So what I'm trying to say, a person, for example, having a B blood group. What blood group? B blood group. He will have antigen A, B. He will have antigen A. If a person having a AB blood group, it will not have any antibody. Like it won't have any antibody. And if a person having a O blood group, it will have antibody of A and B, both. Are you getting my point, Miraculin? Ira?
Yeah.
Yes or no?
Yes, sir.
Now, if a person having a if a person of blood group A got an accident, so from whom he can get blood?
The person who has A blood group.
A person who have A blood group. Anybody else?
O.
Oh,
O. Why O? Because O doesn't have any antibody.
O is a universal donor, right?
Wait, the sound is very bad. Say it
So
again.
O is a universal donor, right?
Yeah, O is universal donor. We will explain why the O is universal donor. So
So
person,
it'll be because they don't have any antibody.
yeah, it doesn't have any antibody. No antigen. It doesn't have no antigen. Okay. Now, for example, a person who having a B blood group, he got an accident. So from whom he can receive that blood?
Sir,
Yeah.
can A group also receive blood from AB?
Okay. So a person having a like AB blood group. So AB blood group has antigen A as well as B. Now B is the antigen which will fight with the B antibody, and it will die. So the blood will get waste. There is
But
no
it will
use
get
of that
A,
blood.
right?
Huh?
But it will get A.
But the both the cell will die. No, the whole cell will die. Whatever the cell you will give, the AB, it doesn't will only the B will go AB, you go outside. It won't be like this. The whole cell will die. Okay. Now second one, if a person having a B blood group, how from whom he can get the
B
blood?
and O. B and O.
B and O. Why B? Because B have the antigen antibody A, so no problem, and O doesn't have any antigen, no problem. Now, if a person is having a blood group from of AB, so he can receive from whom?
O. O.
He can receive from O, and
He can receive
because he
from
have no
everybody.
antibody.
He can receive from
Yeah.
everybody.
Yeah, he can receive from everybody like A, B, AB, and O.
So it can't, but it can't donate to anybody. Like,
Can only
Yeah.
donate to B.
And if a person having a O blood group, from whom he can receive?
Only O.
O.
Only O. Why? Not from A, not from B, not from AB. Why? Because A and B both antibodies there.
Yeah.
That's the reason. If a person who is a donator, so like A can give blood group to whom?
A.
A.
A and
AB.
AB. Like he can donate to AB also because AB doesn't have any antibody. There won't be any problem. A person who's
So
having
like
B
it
blood group,
depends on. So it depends on who has the antibody, right?
yeah.
But doesn't O has AB antibody?
If a person who have AB antibody, he can donate. Like he has no antibody, so he can donate to, like O can donate his blood group to A, AB, B, and O. Everybody. Okay. And he can give blood group, like AB person can give blood group to AB only. Everyone got my point?
Yes, sir.
Did you understand this thing, this concept?
Yes, sir.
sir.
Yeah.
So the table is already given in chapter 22, sir.
Yeah. Now you can make this. Or you can take screenshot after the class, you can make it.
Yes, sir.
Did you understand?
Yeah, sir. Today's science class was interesting.
Yeah. So this is what the thing. Now we will understand the next concept tomorrow. Not tomorrow, on Monday. That IA and IB is there. Then what we have to do? Like a person having a blood group, a parent having a blood group IA and IA. Then what will happen? Like this. Okay, if you understand this, then we'll end this class. We did the sex determination in human beings, how it happens. I explain it to you. And this also we did it. We'll do some more, and by Monday we'll complete this chapter. And Monday we can start the new chapter also. Okay, so we'll end this class. I hope you enjoyed the class. Till then, bye. Have a nice day. Signing off to Shabakir. Ready for the test. Bye bye.
So the test is impossible difficult, you know.
Yeah, impossible to difficult.
Please don't make it impossible. Just make
Like,
it like
Then
on
how can we make it